everyone. I'm Probodh Satyal from University of Alabama-Huntsville, and today I'm going to present bioactivities and chemical composition of Himalayan aromatic and medicinal plants from Nepal. The topic looks very broad, but today I'm going to pick out just only one species genus that I have done. I have done essential oil from Himalayan grains, and I have seen their biological activities. And for that project, we tried to lay foundation of chemodiversity in the Himalayan range, and we tried to determine their possible therapeutic and medicinal use. And for that project, we had collected 100 more different essential oil and 18 more different biopsies were performed in those essential oil in the collaboration of Nepalese students from Tribhuwan University and from the University of Alabama-Huntsville. We performed that project. And probably you may know who is Nepal. Nepal lies in between India and China. It's a landlocked country. And the alternative identity of Nepal is Mount Everest, the tallest peak of the world, which has 8,848 meter height. And this is Topographic Map of Nepal. On looking, it is explicitly clear on looking over the Topographic Map of Nepal that it has altitudinal variation, so there is the highest chances of having many more species. And on looking over the climatic condition and the number of medicinal plants available in Nepal, Nepal is the best site to do site to source for natural product research. And today I'm only going to discuss on Artemisia species, Artemisia genus that I have collected from Nepal. And I collected 3 Artemisia, Artemisia dubia, Artemisia vulgaris, and Artemisia indica from different places of Nepal. They were around 2,000 meters from the sea level. And one was from the Kirtipur, and another was from the Khabre, and another one from the Hetoda. Those were the local places of Nepal. And Artemisia belongs to Astraceae family, and in Artemisia there are around 350 species of aromatic shrubs and herbs, and it is distributed all over the world. And it has been traditionally used in various medicinal purposes. And it is also a good source of commercial essential oil. And we collected the plant material, and we did Clevenger type hydrodistillation and collected the essential oil. And the essential oil was injected through the GCMS, gas chromatography mass spectrometer, to get the essential oil component present on that essential oil. And on the mass spectrometer, we got the retention time and the mass spectra. And the retention time is converted to retention index by using those formulas at these times. And we compared the retention time and the mass spectra with the literature from this book. And we also used niche database to get the composition result. And this is the summary of our composition. And in the Artemisia dubia, the major component was chrysanthemum and camphor and coumarin. Likewise, in the Artemisia indica, isoascaridol and the ascaridol are the major components. In the Artemisia vulgaris, alpha-thuzone, beta-thuzone, camphor, sinew are the major components that were found in those species. This is Artemisia dubia, and the major component was chrysanthemum 29%, camphor 16.8%, coumarin 18.3%. I have tested the major components for bioactivity also to see the synergistic effect. Chrysanthemum was not commercially available, so I was unable to perform biological activity with chrysanthemum. Just I did camphor and the coumarin. And the plant is traditionally used to treat cut and wounds, and the plant part is also used to treat various digestive problems and for the ringworms. And I studied another plant from Artemisia, which has the major component as ascaridol, and the isoascaridal, those were also not available commercially. And the plant part is used to treat ringworms, skin diseases. Those were the traditional use. And this is Artemisia vulgaris. This is one of the major essential oil produced all over the world. And it has the major component of alpha-beta-thusin, 1,8-cineol, and the camphor. I performed biological activities of all of those three components. And the plant has various medicinal uses to treat intestinal worms, to treat digestive problems. It works as insect repellent, and in treating nosebleed and neurological disorder, they are useful. And it is also used in the manufacturing of incense. And it has some allelopathic activity. Allelopathic activity, I will discuss it later on. And Artemisia vulgaris were studied in various parts of the world, and we did cluster analysis of that. Our composition with the all of the world's composition, there were around 28 compositions compared, and we made that cluster diagram. And Nepal's chemotype was found to be alpha-thusin, which is similar to Turkey and the India sample. And in the Artemisia indica, you have seen three components came together: ascaridal, isoascaridal, and the alpha-terpinol. And we thought that, how did it come? It might come from some hydrodistillation or from the GCMS thermal temperature. And we tried to figure out why it came by using theoretical chemistry. We use Spartan software to see the reaction. This is ascaridal, this is alpha-terpinol. And this is retrolytical reaction, whether they come from retrolytical reactions or they came from, this is thermal rearrangement by free radical mechanism. And ascaridal, this ascaridal's isomer is isoascaridal. It can also, we thought that it could have occurred by some thermal process, and we tried and see, and we found that delta Z value seems to be positive, that is Gibbs free energy. If Gibbs free energy is positive, then the reaction is not spontaneous. So we thought that they came from some other sources, some other biosynthetic pathway like chemical acid, muvalinic acid pathway. So these are not the reaction happening during hydrodistillation or by the GCMS temperature. And we did cytotoxicity on the breast cancer cell, on the MCF7 cell by using MTT assay. And the cytotoxicity results tells us that A dubia is the most active, it kills 100% cells, whereas Artemisia indica and Artemisia vulgaris were in the various amount. And I have correlated that with the result with the major component. Artemisia dubia content camphor and coumarin are the major component, and they have some good activity with this cell line. Camphor kill 86% and coumarin kill 46%, and it could have occurred due to synergistic effect. And we did the antimicrobial activity in the Brine Shrimp, antimicrobial activity in the microbial dilution technique. And the result is in favor with the A indica and the A dubia. Those were the most active, but A vulgaris were not active. It is the weakest one. And its major components, alpha-beta-thusin were tested. And it was found that alpha-beta-thusin was not active for the microbiological activity. So we concluded that is inconsistent with the result. And Artemisia dubia and Artemisia indica were showing marginal activity for the microbial activity, and it was due to presence of chrysanthemum, coumarin, and the ascaridal and isoascaridal. And we did the allelopathy activity of those essential oil against two kinds of seed, rye grass and the lettuce seed. Allelopathy is the inhibition of growth of plants in presence of release of any chemical from other neighboring plants. Like that here, the chemical is released by this plant and their growth is suppressed. This effect is called allelopathy. And we did allelopathy in the petris and we observed their growth. And the results looks like that. Artemisia vulgaris is best for the lettuce seed, whereas A indica did not have, it has lesser activity. It has less ICP value. And I have correlated this with the major component present on that essential oil. The major components were major component in vulgaris. You can remember those were Tribhuwan and the camphor. They also have good activity. So we can correlate the bioactivities of A vulgaris with their components' biological activity. And likewise, we did that in lettuce seed also. In the lettuce seed, the most active was found to be Artemisia dubia. Artemisia dubia contains coumarin, camphor, or the major components. They also have good activity. So the result is inconsistent with the its major components result. And we also did nematicidal activity. Nematodes grow in the soil in presence of some bacteria. They come to the food and they go through the intestine, and they can disturb your CNS. They can disturb your central nervous system, and they can cause your heart failure. So they are very harmful. So we tried them on essential oil, and the result is in favor of the Artemisia indica. That may be due to ascaridole and isoascaridole, which has been already studied in various literature that ascaridole and isoascaridole containing sample have nematicidal activities. They are antiparasitic, so it is inconsistent. We tried with fire ant. We tried fire ant on the beaker. I think you might have ever experienced the biting of fire ant. That's the symptom of fire ant biting. And we did a fire ant insecticidal activity. And it shows that A vulgaris is the most active one. And we tried its components, Tribhuwan and the camphor. They are also active one. So we thought it is inconsistent with the result. And we did fire ant repellency test also. In the fire ant repellency test, we did them in the two tips joined together, and there was a small hole, and in one tip there was the control, and another tip there was the sample material dissolved in the clean water. And after 2 hours we counted them, and the repellency test was like that. Artemisia vulgaris was the most active one. And its components camphor and the Tribhuwan has also good activity. So it is also inconsistent with this result. And we also do the fruit fly bioassay. Fruit fly is the disease carrier. It can carry various diseases, and we rear them in that kind of tubes. We purchase them from Carolina Biological Supply, and the results is in favor of A vulgaris. Looks like A vulgaris is good for insecticidal activity. And we did a glassworm also. In the glassworm, Artemisia indica was active. It is antiparasitics. It is also showing the same activity over here. I also tried Artemisia salina. In Artemisia salina is for the toxicity test, and it has shown good activity in the Dubai, which we have seen in the cytotoxicity also. In cytotoxicity, Artemisia Dubai had killed 100% of cell lines. It is in correlation with that result. And the conclusion of my research is that the essential oil from Nepal have shown wide range of bioactivity. Artemisia vulgaris is good for insects. Artemisia dubia has shown comparable antimicrobial activity. Artemisia indica has shown antiparasitic effect. All of them exhibited good allelopathic activity. And by looking over their components results and the essential oil results, we found that there is the presence of synergistic effect in essential oil. Essential oils are stronger than its individual components. And essential oil can replace harmful chemicals, pesticides, which are environmentally unfriendly. We can avoid them. And our result is consistent with ethnobotanical uses also. There is a huge potential in the Himalayan range for discovery of new natural product compounds. And those were the references that I used. And I will acknowledge my professor, Dr. William N. Setzer, for helping me during this project, and Dr. Bernard Bogler for technical assistance, Dr. Lynn Boyd for nematicidal activity, and Mr. Probodh and Ambika Powell for preparing the slides. And the last conclusion of my project is that essential oils are truly essential. You can use it in all purposes of your life. You can prepare it lowest cost. You can prepare that in your home also. You don't need any harmful chemicals. Avoid chemicals, use only essential oil. That is my last sentence to you. Thank you very much. Do you have any question?
There's time for one question. Is there one question?
Yeah, one question I have. Yeah, just it's a very, very nice presentation you have done. Just I want to know very, very small questions. I mean, medicinally valuable you have mentioned there. Which one is the most valuable? I mean, for the medicinal.
Out of these three, or out of all of my research?
Yes, yes.
I found Cinnamomum glaucescens is the most active one in all of the biological activities that I have ever studied.
Okay.
Yeah, Cinnamomum glaucescens.
Okay.